Hello guys and welcome back to another video. In this video, I'm gonna show you more about the controls lab as well as lab 1. It's gonna be like a walkthrough. So let's start. I'm gonna start with the first lab, which is the controls training lab, where all of the controls are shown.
Welcome to
Let's start.
MicroVlabs training lab. My name is Ruth, and I will guide you in performing this lab. Let's start by learning basic controls.
Well, let's start by learning basic controls.
Follow me. Use the WASD or arrow keys to move around.
So you can use the WASD
Excellent.
or the arrow keys to
Use
move
the mouse to
around.
look around. Excellent.
You can
To
use the
interact with
mouse
an
just as you
object,
would play
point
another
towards it
video
until it
game.
highlights.
It's asking me
Point
to
towards
point
the loop until it
towards
highlights.
an object until it highlights. So let's do that.
To perform an action, you can use shortcut keys.
Over
or
here, the
action wheel.
video pauses a
list
bit.
of possible
Sorry, the
actions and
gameplay
shortcut
pauses a bit.
keys is displayed at bottom left
Don't
corner of the
worry.
screen. Click the left mouse button once to pick an object.
So you can click on the button, the left mouse button, to pick it up.
Brilliant. You can use shortcut keys or an action wheel to interact with an object. This time, let's use the action wheel to pick up an object. Press and hold the spacebar button.
So again,
to see
the
action.
gameplay is paused,
Move the mouse
so you
over
can't
an
do
action,
anything right now, so
and
don't
let go
worry.
of the spacebar to perform the action. Let's try this.
Now it's asking you to start. If you get stuck over here, you can again restart, but let's do it. Just press spacebar, use our mouse to go over the action, and then just let go the spacebar. Don't click anything.
Great.
There we go.
You are getting the hang of this. After picking up an object, you can place them down or put them at specific places. Let's try this. Use the left click to place the loop down anywhere.
Let's place it down over here.
Now pick up the pipette.
Let's pick up the pipette.
Press shift and left mouse button to put the pipette in the tray. Let's try this.
So you can point towards the tray, or if you do just like a click like this, it's gonna fall wherever you want it to fall. But if you press shift and then do the click, it's gonna go back in the tray.
Let's try this with the action wheel. Now pick up the pipette.
Let's try this with the action wheel. I'm gonna pick it
Point
up.
towards the tray and use the action wheel to place pipette in the tray. Let's try this.
So I'm gonna point towards the tray and then
Brilliant.
place in tray.
Now let's learn how to interact one object with another. Example, how to latch a tip on the pipette, or how to aspirate or dispense media. Let's try this. Now pick up the pipette.
So I'm gonna pick up the
Point towards
pipette,
the tip box.
point towards the
X
tip
or
box.
use the action wheel to open the tip box.
So sometimes it says to point towards the tip box, but you might be a little bit further away, so you might go a bit closer, but don't go too much close. Uh, Otherwise you might miss. But as soon as it highlights, you're good. You can press X.
Press 1 or use the action wheel to latch a tip.
Again, you multiple options. If you just keep forgetting which to get, you can just use get tip. There you go.
Excellent. Press P or use the action wheel to spawn more tubes.
So I'm gonna press P.
Let's add media to the empty tube. Point towards the flask with press X or use the action wheel to open the media flask.
Okay, I'm gonna use X to open the media flask.
Press 1 or use action wheel to aspirate media.
I'm gonna press 1.
Now point towards the empty tube.
I'm gonna point towards the empty tube.
Press X or use the action wheel to open the tube.
I'm gonna use the action wheel this time.
Press Q or use action wheel to dispense media in the tube.
I'm gonna press
Great.
Q.
You are getting the hang of this. Press G or use action wheel to discard the tip. Well done. I think you've got the hang of the controls and are good to go. Remember, performing an experiment is only possible if good lab techniques, GLPs, are followed. Proper cleanup is an important part of GLP and is equally important as the experiment itself. This ensures the working environment is hazard-free and all instruments, chemicals, and reagents maintain their integrity for the next experiment. So let's make proper cleanup a habit as well. Let's start cleanup for this lab. Let's start by discarding all tubes. Pick up each tube one by one and discard them appropriately. Tubes with media go in the biohazard, and tubes with reagents go in the sink, and empty tubes go in glass waste container.
Over here it gets a little bit tricky. But let's first put the pipette down in the tray. Let's pick up, for example, any tube. Let's pick up this one. So it says to discard in biohazard. So what I'm gonna do is I'm gonna press G, then I'm gonna click on B. So it's gonna discard it in biohazard. Now it asks to pick this up, and for this I don't do biohazard because it's empty. I'm just gonna do sink. Well, if you just click sink, if it's empty, it just goes automatically to the glass container. Again, S. If you get this wrong step, you might not be able to complete it successfully. So it's important to throw the tube with the media in the biohazard and the one which is empty in the sink or the empty container. So now I'm gonna close this. Gonna close this also.
Make sure your pipettes are reset to the maximum value and are in the tray.
So it's set to 200. I can use the plus and minus button to, increase or decrease the pipette value. Gonna place it back in the tray.
Congratulations on learning the basic controls.
I'm gonna look out for other prompts.
Your lab scores depends on the way you perform your experiment, your results, and proper cleanup. You can now submit the lab.
Okay, now let's submit the lab. And there you go. It's done. It's done correctly. So all of my labs are actually unlocked, but if it's done correctly, then you'll see a lab tech. Wait one second, let me show you. You'll see if the training lab was done correctly, you'll see this achievement pop up on your screen that you have unlocked the certified lab tech achievement. And once that's unlocked, your lab 1 should automatically also get unlocked. So let's start with lab 1. Again, let's start the whole training. Let's do complete training.
Aseptic techniques in culturing lab. My name is Stephen and I will teach you 2 commonly used methods in microbiology. In this lab we will learn streak plating and pore plating using aseptic techniques. Let's start. You have been provided with an overnight culture of E. coli. You will use the loop to streak the culture on a plate with nutrient agar. Make sure to flame the loop to sterilize it before using. Next, transfer 100 microliters of the culture to an empty plate for pore plating. Bring in nutrient broth deep from the water bath and let it cool to roughly 55 degrees. A word of caution, nutrient deeps are at 99 degrees in the water bath. Make sure to wear proper protective equipment in real labs. Once sufficiently cooled, the media on the plate with 100 microliters of culture and wait for the media to solidify. Place both plates in the incubator and incubate for 24 hours at 37 degrees Celsius. Once colonies are visible, place both plates on the results table. All plates that you submit with the lab will be stored in the 4 degrees fridge and are available in subsequent attempts of this lab. You can view all main objectives of this lab on this board. At any time, press the escape button and use the menu bar to repeat instructions, read the lab manual, or submit the lab. Good luck! Streak plate is a simple technique where an inoculum is taken on the loop and streaked across the agar. After incubation, bacteria can be seen on the surface of the agar. To perform streak plate, we need to first sterilize the loop.
Okay, so we're just gonna pick it up
towards the flame and press F, or use the action wheel to flame the loop.
So again, point and just press F.
Use the action wheel, or press Shift plus left click to put loop in loop holder.
Okay, I'm gonna do Shift and click. There we go.
Let's get the nutrient agar plates for streak plating.
Okay. Press P. Pick up the plate.
Pinch zoom. or mouse wheel to move an object near or far for a more precise control.
You can use pinch zoom and place it over here.
Let's get an empty plate for pour plating.
Okay, let's get an empty plate for pour plating, and there we
Wait
go.
for the loop to cool down, and then pick up the loop.
It's gonna 3, 2, 1, yay, it's
Press
cooled.
X or use the action wheel to open the tube.
So I'm gonna press X.
Every time you open or close a tube, you should always flame the opening to minimize contamination from air.
Pressed F.
To get bacteria
Again.
on your loop for streak plating, you just need to dip it once or twice in the culture, point towards the open tube and press Q, or use the action wheel to get bacterial sample.
So let's press Q.
Open the plate with nutrient agar for streak plating.
So again, point towards the plate, press X.
Holding the loop, press Y or use the action wheel to go to inoculate mode.
I'm gonna press
Press
Y.
and hold the left mouse button and drag the mouse across the plate to streak bacteria.
So this is important. So you press and hold the mouse button and drag across.
Press Y or action wheel to go back to lab mode.
So I'm gonna press Y to go back.
Part is done. You can now close the plate. It
Okay.
is important to label your plates properly. Make sure to include important indicators like name of bacteria, media, date, and your initials.
So I'm gonna label the plate. I'm gonna label it as streak plate for now. But all those recommendations that are listed over here are very important in your actual lab.
The general idea behind pour plate is to mix bacteria evenly within agar. One way to accomplish that is by adding a volume of bacteria to the empty plate and add molten agar on top of it. Once the agar solidifies, it will have the inoculum spread evenly within it. After incubation, bacteria will be visible growing inside the agar. Pick up the 200 microliter pipette and attach a new tip.
So pick it up, press X to open, and then 1.
Microliter of bacteria.
So it says to take 100. This is 200 right now, so I'm gonna switch it down to 100. So the plus and the minus button on your keyboard, you can use those. So now it's 100. So I'm gonna press X to open it. Then I'm gonna press 1, 2.
Open the empty plate for pour plating.
Okay, I'm gonna open it
While
up.
holding the pipette, press Y to go to inoculating mode.
You can't go in the inoculating mode unless you're not holding something that can inoculate, like a loop or a pipette. So while I'm holding this, I'm gonna press Y.
Plate to add liquid inoculum.
So I'm gonna add some liquid inoculum right at the center of the
To
plate.
lab mode, and place the pipette back in the tray.
Okay, so first I need to discard the tip. I'm gonna press G to discard the tip, and then Shift plus left click to put it back.
Nutrient agar deep from the water bath.
I'm gonna go to the nutrient water bath.
Deeps are very hot. Make sure to wear proper PPEs while handling them. Take the deep back to your workstation and wait for it to cool to 55 degrees.
So
Deeps in real labs may not cool as fast and may take 5 to 10 minutes based on lab climate.
again, it's important to note that these deeps, which is very hot right now, may not cool as quickly in your real labs. And again, there are more ways to do this. You can add liquid inoculum directly to the plate or to the tube once it's cooled down. Both will work, and you're free to do whichever method you choose. And just press X to open, and then point towards the plate and press Q
Success
to
in pour plate
dispense.
depends on temperature of the deep. If the deep is poured in hot, it will kill all the bacteria in the plate. If you wait too long, the agar will clump or solidify in the tube. A general rule of thumb is, if you can touch the deep with your wrist for more than 5 seconds, it is cold enough to pour. Label your plate.
So this actually does happen a lot in the labs. Students or users actually do end up either waiting too long to pour their deeps, and they get clumps within the plate or within the tube, or what they do is they add in too hot and they basically kill all the bacteria. So it's important to wait for the deep to cool down, and a good way to test is if if the butt of the tube, you can hold it over here for like 1 or 2 seconds without getting an ouchie. You're good to go and you can pour it right away. But it's a good way over here to visualize this experiment. Like you can try pouring it in too hot, or you can try waiting for it to cool down to see what those outcomes are as well. For now, let's close the plate and let's press L to label it. And we're gonna label it pour plate just for simplicity right now.
Pick up the
Update.
street plate and put it in the incubator. Walk towards the incubator with the plate for the incubator door to open.
Okay, so I'm gonna
Put the plate in the
place it
incubator.
in the incubator. I'm gonna place the other plate also in the incubator.
Close the incubator.
Close
Press
it.
5 to incubate for 24 hours.
The best part of this software is just press 5, bippity boppity boo, 24 hours gone.
Pick up the street plate and move out of the ink. Press the N key to observe the colony morphology
See,
from top
so
side.
this was how I plated it. Gonna observe it from top side also.
Press I to view incubation time and colony counts. Press I again to hide the table. You will find details about your plates and tubes and tables by pressing I.
So you have this table over here, and it says that in the street plate there are 129 colonies. The incubation time was 24 hours. In the pour plate we have 61 colonies. Those which are called add label are in the fridge, so don't confuse with that. So in the ones that I made today, we have 129, and again we have 61 in the other one. So I'm gonna place it, hide
Place the
it.
street plate on the submit results table.
Okay, I'm gonna place it over here.
Place pour plate on the results table.
Make sure this green box appears every time you place a thing on the results table. Otherwise, if it doesn't, then it's out of the bounds for the result, and you might not get marks for it. And again, see the difference between streak plate and pour plate is that when you do pour plate, the bacteria is growing within the agar, not on top of it. And you can see that all the colonies are not perfectly round. They are different size, basically the agar restricting growth from each sides. You can also see the objective tables. It's highlighted. You can also press O to see both objectives are done. Press O to hide that. And I'm gonna press pour. I'm gonna place
Make
the
sure the lab is clean
plate here.
before submitting the lab. Press tab to submit lab.
So you won't always get these prompts that what needs to be done, especially in the higher labs, because of course you need to know how to do proper cleanup once you are in the higher labs. But it's a good way to learn, right? What needs to be done. You need to sterilize the loop. I'm gonna just press F, shift click back in the holder. I'm gonna reset pipettes to the maximum volume. This is very important in real lab, not as much in virtual, but because again it's just a virtual. But in real labs, you should always set your pipettes back to the maximum volume, so their spring doesn't get overburdened. This enhances the pipette life. You need to close this, of course, and discard all tubes that were in that you used. So before I discard this tube, I'm gonna tell one more, a few tips and tricks again. So if you notice that something is not being highlighted or something is not doing its function, you can always just look away and then look towards it again, and you will see that it's doing that function. It just resets everything. Again, if something, let's say, gets stuck behind a wall and it's not coming back, you can hold the right click, and to force it back. So these are just glitches, because again, no real computer software is perfect, but try to identify all the glitches there are, and try to get around those. So again, if something gets stuck, it's not moving at all, you can always use the right click to force it back. Again, if stuff gets lost from your tray, you can press R to make sure to call everything back. Like sometimes what happens is that your loop gets, whoop, there goes my loop. And what I did was, because sometimes it's difficult to find, just press R to call back. And of course, since this loop fell down and I have to reheat it, you can keep the loop in holder or on the tray, that's fine. So I'm gonna do discard this. You don't have to come here every time, but still. So I'm gonna discard this in the biohazard. It's a little bit shaky, but again, that also depends on which computer you're using. So I'm gonna close this door. Also, this is fine. This is fine. Everything else looks fine. There are no more prompts. So this is basically what you need to do. And I can now go ahead and submit the lab. Just wait. Okay, so again, my streak plating is good, pore plating is good, all tubes were discarded, pipettes and tips, everything good, general cleanup, workstation is acceptable. So I hope you guys can, follow this tutorial and get perfect scores as well. And if you note any problems, just feel free to reach out either in the comment section or just email me, and I'll try to fix any mistake or any bug that you found out, or try to guide you an alternate way. So that was a nice little walkthrough of our controls lab as well as our lab 1, and I hope you guys really enjoyed it. You found it useful. You can use this tutorial to get full marks in both controls lab as well as lab 1, and I hope you guys really enjoyed it. So that's it for this video. Let's see you in the next one.